Welcome everyone to week number 3. Okay, this is like the 4th time I've started over on this video. I have no idea what's going on with my computer, but it is a roller coaster ride today. So here we go. Week number 3. We have classification of life in cells. Let's get going. See if I can get through it this time. Last week, sorry for my voice. I've still got crud left over from COVID, so I'll be clearing my throat. Last week we learned about the chemistry of life, so actually it's biochemistry. We learned about the importance of water, the fact that it absorbs heat from all the chemistry that's going on in cells, to be a solvent that things are dissolved in, to be an adhesive, it sticks to things, to be cohesive, the water molecules stick together, and that's really important. So it is absolutely necessary for life, and that's why when we search for life out in the universe, we're always looking for the presence of water, because that will tell us whether there's a possibility that life exists there. Then we learned about carbon. Carbon is the element of life. And why is it important? Because it forms 4 bonds with other atoms and molecules, and therefore can make very, very large molecules and very complex molecules. And that's what is needed if you're going to make the variety of organisms that there are in the world. So that's very important. And then you learned about the organic molecules that we call macromolecules, which just means big. Carbohydrates are used as energy molecules. Everybody knows that, right? Everybody in the class is probably on a low-carb diet right now. So anyway, carbs carry energy and are quick sources of energy. They're also used structurally in cells. Then we've got lipids. Lipids are hydrophobic. They repel water. A lot of lipids are energy stored in the form of fats, but then we also have waxes, which help to waterproof things, and whole groups of lipids are hormones, which are chemical messengers that travel between things, and especially between cells and multicellular organisms. And then we've got proteins. Most of the proteins are enzymes, and enzymes catalyze all the reactions that happen in cells, and they carry out the work, the chemical work that's happening in cells. So they're extremely important. Some proteins are structural. Then we have the nucleic acids. A few of those are energy molecules like ATP, adenosine triphosphate, which is the most important energy molecule in cells. But the others, like DNA and RNA, are the molecules of heredity. So they are also extremely important. We will be using these terms throughout the rest of Biology 120, so don't forget them, okay? They're very important. The topic for this week is classification and also cells. So classification is putting things into categories. And why do we do this? We do this naturally as human beings. We put things into categories because that way we don't have to learn over and over and over and over again what a chair is every time we come in contact with a new chair, right? If we know the basic characteristics of a chair and what the basic function of a chair is, the next time we see one that we have never seen before, we know what to do with it, right? Which is a silly example, but it shows you how our minds work. So scientists use classification too, and we classify organisms all the way from domains. That's the 3 biggest categories of life on the planet. Eukarya includes all the eukaryotic cells, which I'll mention again in a minute. Eubacteria contains the true bacteria, and archaea, the 3rd domain, contains all the archaebacteria, which means ancient, but they're really not as ancient as the eubacteria. Anyway, there's just a different category of bacteria that live in very extreme environments. So we have classification like that all the way down to the species level, which includes all the organisms that have very, very similar characteristics. And in some of the species, the members of the species can interbreed. We can't always use that part of the definition. If you're talking about bacteria, bacteria don't interbreed. They've just split in half and become two organisms. Yeah, there's no interbreeding with bacteria. Okay, you need to read chapters 20, just that first section in chapter 20, to give you an idea about classification. And it's good. You don't have to read it in detail. You don't have to memorize phylum, class, order, family, genus, species. You don't have to memorize that, but just read through it so you have a general understanding. And then read through chapter 4. You're going to need that information because that describes to you the two different basic types of cells, which I will get to in just a minute in one of your assignments, a description of one of your assignments. Remember that OpenStax textbook, Biology 2nd edition, is free and online. Please, please use it. Please don't just Google this, because there is so much garbage out there on Google, isn't there? I mean, oh my goodness, there's just so much. So anyway, use your textbook. There's a table of contents on the left side. You can go directly to these sections over in that table of contents. So it is extremely easy to use. All right, read the Module 3 overview and watch the videos. The Invisible Universe, which talks about the human microbiome, which is very, very important. So we don't know everything that we need to know about the human microbiome. And so what the heck is that? That's all the microorganisms that live in and on us and should be in all these different locations in and on our bodies. Let me tell you one little story. So I had a student when I was teaching at the college in North Carolina, and he ended up being diagnosed as a paranoid schizophrenic, which was very sad because he was in his early 20s. Well, he had gone home and told his parents that he had gotten an eye infection from the E. coli bacteria that we were working with in microbiology. And it got to administration, including the president of the college. And here they come after me, marching. Janie, this student got an E. coli infection in his eyes from your microbiology class. I said, okay, let me explain this to you. E. coli are found in your large intestine. E. coli cannot infect your eyes. I have never heard of one single case of this. E. coli can't survive in your eyes. They only survive in your large intestine, nowhere else. Not in your mouth, not in your ears, not under your arms, not in your belly button, not anywhere else. He cannot have an E. coli infection in his eyes. And so that was put to rest. But that's part of this microbiome that we need to understand better. We need to understand which microbes we need, how many we need, which types we need, and where we need them. We have got all different kinds of ecosystems around our bodies. The bacteria and other microbes that are found in our armpits are totally different from the ones in our belly button. And you may laugh that I'm saying belly button, but there's a whole group of scientists who study nothing but the microbes in your belly button. I'm not kidding. As compared to your colon, compared to your skin, compared to your mouth. You have all these different ecosystems around your body. And this is a very active area of research because we just do not understand everything that we know about them. And I know that people take probiotics. I sure do when I take antibiotics. But we don't even understand that, how to replenish the microbes in our colon after we wipe them out, kill them with by taking antibiotics. So anyway, the Invisible Universe is important. Domains and Kingdoms of Life. Yeah, watch all the videos there. They're very short and they are informative. I'm also going to post this extra video because it is absolutely astounding and amazing. It's a Harvard study. It's an antibiotic study that shows you how quickly antibiotic resistance happens. You expose bacteria to antibiotics. Some members in that population are going to have natural immunity to that amount of antibiotic. Not only that antibiotic, but that concentration. And this study shows you how quickly antibiotic resistance occurs. This is a very short video clip, and it is explained to you how this works. Take a look at it. Use this in your work this week, especially when you're explaining about antibiotic resistance, because it is phenomenal how quickly this occurs. By the way, this is a picture of a sea eagle in Scotland when I went in May. I was standing on a boat. It was rocking because the seas were very bouncy. It was rocking, and this eagle was hunting the puffins that were all on those rocks that you see right there. And somehow, I got some good pictures of it, and I was very proud of myself for this. Sea eagles are very, very difficult to get pictures of because they usually circle way, way, way over your heads, but this one was actively hunting for puffins, which I love, which I'll share pictures with you during the term. But here's a sea eagle. Next week, if you watch my video, let me see if I can do this. Hello, wake up. I'm going to share with you that word respect back there. I'll tell you the story about that. Okay? All right. This week, what do you have going on? You've got your discussion board postings. And these are the questions that you're going to be answering. Why antibiotics target? So you take antibiotics, which are drugs, which are basically just chemicals that kill the bacterial cells in you, not the viruses, not the fungi, not anything else, just bacteria. Antibiotics target bacteria. So antibiotics kill bacteria, which are prokaryotic cells, but they don't kill your cells, and that's how they can cure you, because they select, those chemicals select the types of cells that they kill. All right, so here's the trick. Bacterial cells are prokaryotic cells. They are very tiny, very simple. Every molecule in the cell comes in contact with the other molecules in the cell. This is kind of like a tiny home, right? If you've ever seen pictures of tiny homes, you open the door, and there's the bed, and there's the kitchen, and there's the table that you sit at to eat, and there's your closet, and there's the bathroom, right? Everything is in contact with everything else. You can see everything in your whole house just if you stand at the front door. That's kind of like a prokaryotic cell. Then, our cells are eukaryotic cells. We have all these little compartments in our cells. We have mitochondria. We don't have chloroplasts, but plants have chloroplasts. We have peroxisomes. We have nuclei. Oh my goodness, we have the endoplasmic reticulum, the Golgi apparatus. We have lots and lots of little compartments in our cells that have specific jobs. And this would be like, if you've ever watched The Real Housewives of New Jersey, one of those houses where they have so many rooms that you don't even know what the rooms are for anymore when you go through the house. That's kind of like our cells. Our cells are much more complex. They have lots more going on. So that gives you an idea that there are huge differences between prokaryotic cells and eukaryotic cells. And that is true. So antibiotics have to target something that's different in bacterial cells than in our cells. I'll give you another hint. To start with, look at ribosomes. That's really important, a really important target. So now you got to figure out what a ribosome is to see how antibiotics could target ribosomes. So take a look at that. That's going to be in chapter 4. You're also going to explain how our use of antibiotics increases antibiotic resistance. So people are not antibiotic resistant. You and me. Because when we take antibiotics, they don't kill us. So we can't have resistance. But the bacteria become antibiotic resistant. And that's another reason to watch that Harvard video that I'm going to show you. What is it that we do in the way that we take antibiotics, in the way that we're prescribed antibiotics, in circumstances where antibiotics are prescribed? How does our use of antibiotics increase the rate of antibiotic resistance? It's always been said, as soon as you start using an antibiotic, some bacteria are becoming antibiotic resistant. But how fast they come resistant is up to us and how we use these antibiotics. So take a look at that. There are lots of superbugs out there now. Man, I had an infection by one 2019, so that was nasty. But like MRSA, methicillin-resistant Staphylococcus aureus, I can bet that you've heard of that. Those staph bacteria are very resistant to lots and lots of antibiotics, not just methicillin. And then in your response post, you're going to discuss the use of antibiotics in livestock and chickens and turkeys. I guess livestock are considered, turkeys and chickens are considered livestock. I don't know. But since the 1950s, farmers have been giving antibiotics in livestock food to farm animals. And that's because it increases their muscle mass, it increases the size of eggs, it increases meat, the amount of protein that's there. And that's because the antibiotics fight off all these silent infections that these animals have. Plus, it keeps the livestock and all these different farm animals healthy longer. They don't get sick, even though they're all crowded in together. But it is creating significant problems, all this use of antibiotics in livestock. And in fact, I think the statistic is that in this country, 80% of our antibiotics are fed to animals, not given to human beings. So that gives you an idea of how widespread this is. You're going to start your final project. I'm going to explain this more in just a minute. You're going to choose your topic first, and several of you have emailed me and want to choose different topics, and that is wonderful. I love that. Use the guidelines and rubric. So what the heck does that mean? Guidelines are instructions. Rubric tells you how I'm going to grade it, because I look exactly at the rubric, just like you see it, and decide which category you're in. Are you in the excellent group? You've covered absolutely everything. Are you in the good group, where you covered everything pretty well? You did a good job. Maybe you could have been, you could have tweaked it a little bit more and added a little bit more on down the line, right? That's the rubric. Use the correct format, and I'll explain that again in a minute, because this first final project assignment needs to be an outline, but with details, okay? Definitely not an essay. This is not an English class. I'm so thankful for that. But in science, we really get to the point. I can tell you that for sure. Even when I was taking biology classes when I was in college, some of my classmates, we'd get out of an exam and they'd say, man, I wrote 10 pages. And somebody else say, I wrote 12 pages for that exam. And I'm looking at them going, I only wrote 5 pages. I'm going to fail this exam. I went to my instructor, Dr. Davis. I said, Dr. Davis, I only wrote 5 pages for this exam. He said, Janie, stop worrying about it. You get to the point. You don't BS all over the place. I said, okay. Oh, and that's what you're supposed to do in science. Get to the point. Okay, let's take a look at milestone 1. That's what's coming up. Guidelines and rubric. Here's your list of topics. I went through this last week. All these different topics. If you want to choose anything besides what's on this list, you must email me and I will approve it, or suggest modifications, or say, no, you can't do that. We're not getting into astrology and what the Scorpio sign means. That is not biology. Okay? But we can figure out something. I tell you, I usually can figure out some kind of connection to biology. So there's your list of topics. But let's look down here at this bottom part, because this is what you have to do. Now, you want your sections labeled. You want to have a Roman numeral 1 that says introduction right there, right? You label it like that. This is not an essay. So make it in outline form. And this is going to come in handy as you put all these sections together to make your presentation on your topic. Presentation will be a set of slides. So your introduction. Okay, let's look at that for just a second. Describe. Oh, that's a very important word. Describe the topic you selected. So, for example, deforestation. If you start off and you say, deforestation is a very important problem in the world. What is it? What is it? What is deforestation? Don't assume that anybody knows what it is. And maybe you're just saying this because you're not sure what it is. So this is where you start doing your research. Use that Shapiro library that you have access to. It is a wonderful resource because you have access to so many different books and articles and news articles, etc. through this fantastic Shapiro library. Describe the topic. Why is it significant or interesting to you? Why did you pick it? What's this doing for you? And please don't say you had to. You did not. You did not have to pick any one of those. So why is this interesting to you? Why is it significant or interesting in the general field of biology and the world? In the world. I mean, these topics were chosen because they are hot topics right now in the world. Why? Why? Answer that. Support your response with real-world examples. So if you're talking about deforestation, you are definitely going to be talking about the Amazonian rainforest and probably Brazil. The political climate in Brazil has totally changed, but for a while there, the president of Brazil decided that it was a wonderful idea to increase agriculture in the country by slashing and burning all the rainforest. Totally destroying that precious, precious ecosystem. The political climate has changed now, and so it's going back to the way that it was, and it's protected more now. So support your response with real life examples. Okay, and then your topic. So this is the second section. Got your introduction, then your topic. Where? Where does this happen? Which kind of ecosystem? If you're talking about global warming, then the whole biosphere, the whole component of the earth where you find life, that's called the biosphere, is where this is happening. If you're talking about eutrophication of a pond on your cattle farm, this is only happening in the ecosystem of that pond. So you can narrow down your topic if you want to and make it much more personal. And so you see, your little ecosystem is going to be totally different from the one that somebody who's describing global warming is going to have. And describe the locations, not just list it, describe. Where does it happen? And which ecosystems and biomes are affected? How? How does this process happen? What is deforestation? How does it happen? And really, it also helps to say why. And I sort of gave you an example with the Brazilian president. He wanted to increase agriculture for the country, which is a great, that's a wonderful goal. Feed more people. But the problem is, if you destroy the rainforest, you've destroyed so much more that will never be regained. It's just crazy. Who? Which species benefit from this? If you're talking about eutrophication of bodies of water, this is a process where bodies of water age out eventually. And if you're looking at a pond, well, the algae definitely benefit from eutrophication because they grow in abundance in this pond water in massive, massive amounts. People benefit because they're allowing fertilizer to run off into these waterways. But you got to figure this out. Who benefits from this? Air pollution. You know, companies benefit from air pollution if they don't have to provide clean exhaust from their factories, then that's less expense for them, and their stockholders are happy because they make more profit. So who benefits from this? Which individuals in the human population benefit, like the industries? So who experiences the positive side effects of this topic? Those are the items that you should cover in this first milestone. This is an outline form, which I'm going to cover right here. Outline form. The guidelines tell you how to submit this. So it tells you right there, create an outline. Number 1 is the introduction. Number 2 is the topic. Where, how, and who. Word document, double spacing. It has to be a Word document because you have to put this in Turnitin. It uploads into Turnitin, and that checks for plagiarism, which unfortunately last semester I had a full-blown case of that, like 92% similarity between that assignment and 2 others out there in the universe. So it has to be reviewed by Turnitin. Double spacing, that's for me so that I can read it. I don't care about the point so much. I don't care about the font. Just don't make it goofy. Don't make it pink color on a white background. You know, make it easy for me to read so that I don't go blind. 1 or 2 pages. This is not a dissertation. This first milestone. Include in-text citations. So if you get this information from anywhere other than out of your brain, you need to give that person, those authors, credit, and that's an in-text. That means in the body of your work. Citation, and also have a reference section with at least 3 references. Okay, so make sure you use the guidelines and put this in the right format. All right. A reminder, there's my name, Dr. Janie Sigmon, m.sigmon@snhu.edu. I'm here for you. I will help you. All you have to do is ask, and I will help you. So please, I hope you have a wonderful week. And if you need anything at all, please let me know. Okay? See you soon.